In Colombia's rainforests, a creature smaller than your thumb carries enough poison to kill 10 adults. Its venom is 200 times more potent than morphine. Scientists call it the deadliest vertebrate alive. But what really makes these golden frogs walking weapons? The answer is not just their lethal toxin. It is a story of evolution, survival, and a secret that could rival any thriller. So how did something so small become the most dangerous animal on the planet? Barely the length of a paperclip, the golden poison frog lives hidden among the tangled roots and dripping leaves of Colombia's Pacific lowland rainforest. Scientists first described this animal in 1978, but local people had known for generations that its tiny frame held extraordinary danger. Measuring just 5 centimeters from nose to tail, it stands out like a shard of sunlight against the deep green forest floor. Its color is not camouflage, it is a warning. That bright yellow skin is nature's billboard, advertising a threat that few creatures dare to test. The golden poison frog's world is astonishingly small. Its entire species is found only in a narrow band of rainforest in the Cauca and Chaco regions, an area so humid that the ground itself seems to breathe. Here, the frogs spend their days moving boldly through leaf litter and moss, unafraid of most predators. Unlike many amphibians that melt into the shadows, these frogs often perch in plain sight, their color a shield more effective than any armor. Not all golden poison frogs are the same dazzling yellow. In some pockets of forest, populations appear mint green or even orange, but every wild individual carries the same warning: approach at your own risk. Their skin is smooth and glossy, a living surface that holds more danger than any snake or spider in these woods. Despite their reputation, these frogs are not aggressive. They hunt tiny insects, ants, termites, beetles, using quick, precise movements. Males call from the damp leaf litter, their voices rising above the constant drone of rain and insects. After mating, they guard small clutches of eggs, then carry the hatched tadpoles on their backs to hidden pools. For years, the mystery remained. How could something so small, so seemingly ordinary, become the deadliest animal in its forest? The answer lies just beneath that golden skin. Toxicity in golden poison frogs is not something they are born with. The secret is in what they eat. In the wild, these frogs feed on a narrow menu of rainforest arthropods, tiny ants, termites, and most importantly, small beetles hidden in the leaf litter. Among these, mellirid beetles are the leading suspects behind the frog's chemical arsenal. Scientists have traced the same family of batrachotoxins in both New Guinea's pitohui birds and Colombian frogs, and both animals share a taste for these elusive insects. When golden poison frogs are raised in captivity, away from their native forests and fed standard lab diets of crickets and fruit flies, something remarkable happens. They lose their toxicity completely. Their skin becomes harmless and unable to produce the lethal alkaloids that make their wild cousins so dangerous. This single observation changed how researchers understood the frog's biology, and it revealed a simple truth. The rainforest is the source of the weapon, and the frog is simply the carrier. Chemical analysis of wild frogs shows dozens of different alkaloids in their skin, each one picked up from their prey. The most powerful, batrachotoxin, is almost certainly acquired from mellirid beetles, though the exact species in Colombia are still being investigated. In the frog's body, these toxins are absorbed and stored in specialized skin glands, ready to be deployed against any would-be predator. But without their native diet, even the most brilliantly colored frog becomes as harmless as any pet store amphibian. The golden poison frog is a living link in a chain that stretches from the forest floor to the top of the food web. Its power depends entirely on the presence of certain insects, which in turn depend on the health of the rainforest. Remove the right beetle from the system and the frog's legendary toxicity disappears. Here, The ecosystem itself is the architect of one of nature's most sophisticated weapons. Beneath the golden skin of this frog lies a molecule that redefines the limits of natural danger. Scientists call it batrachotoxin, BTX, a steroidal alkaloid so potent that the numbers strain belief. In controlled laboratory experiments, less than 3 micrograms per kilogram of body weight is enough to kill half the test animals exposed. For a 70-kilogram adult, that means a lethal dose hovers around 140 to 200 micrograms. To put that in perspective, a single grain of table salt weighs about 50 micrograms. The amount of batrachotoxin needed to end a human life could fit on the tip of a needle, far less than a pinch of sand. Chemical analysis of wild golden poison frogs reveals an arsenal. Each frog carries, on average, about 1 milligram of batrachotoxin in its skin. That is enough, in theory, to kill 10 or more adults if the toxin is delivered efficiently. These figures are not myth or exaggeration. Toxicologists have measured and weighed the molecule, compared it to the world's most infamous poisons, and found batrachotoxin at the very top. Unlike snake venoms or cyanide, which act by shutting down critical enzymes or blocking nerve signals, batrachotoxin takes a more ruthless approach. It binds to voltage-gated sodium channels, the electrical switches inside nerves and muscles, and forces them to stay open. Sodium floods in, nerves fire uncontrollably, and the body's systems spiral out of control. Convulsions give way to paralysis. The heart's rhythm falters. In a matter of minutes, respiratory failure sets in. There is no known antidote. Compared by weight, batrachotoxin is hundreds to 1,000 times more toxic than cyanide and far deadlier than the venom of any cobra. Its molecular architecture is so precise, so unforgiving, that even the smallest error in handling can be catastrophic. For researchers, working with this toxin is an exercise in extreme caution. One speck, one careless touch, and the outcome is irreversible. The golden poison frog is not just a carrier of poison, it is a living vault for one of nature's most efficient chemical weapons. Inside the golden poison frog, survival depends on a hidden system as complex as any weapon. For years, scientists believed the answer was simple. A mutation in the frog's sodium channels, the very targets of batrachotoxin, would block the poison's effects. But when researchers finally isolated these channels in the lab, the results were startling. The channels from Phyllobates terribilis remained just as vulnerable to batrachotoxin as those from any other animal. Yet, the living frogs were completely unharmed by doses that would kill almost anything else. Even frogs raised in captivity, fed on harmless insects and lacking any trace of toxin in their skin, could shrug off batrachotoxin and saxitoxin alike. The classic lock-and-key model had failed. The real defense was happening somewhere else. The search turned to the frog's blood and tissues. In other animals, like bullfrogs, a protein called saxifilin acts as a molecular sponge, binding toxins before they can reach their targets. Inspired by this, scientists began looking for similar proteins in poison frogs, molecules that could capture batrachotoxin in the bloodstream, neutralizing it before it ever touched a nerve cell. While the exact proteins in Phyllobates terribilis remain uncharacterized, experiments showed that when saxifilin was added to poison channels in the lab, it could rescue them from paralysis. This suggested that toxin-binding proteins, or their molecular cousins, might be at work in the golden poison frog as well. But the defense does not end there. Toxins absorbed from the frog's diet are quickly shuttled away from vital organs and nerves, locked inside specialized skin glands. These glands act as secure vaults, sequestering batrachotoxin in a form that is ready to deploy against threats, but walled off from the frog's own biology. The toxin does its work only on the outside, never allowed to circulate freely where it could do harm. Transport proteins and cellular pumps likely help maintain this barrier, moving alkaloids out of the bloodstream and into storage before they can accumulate to dangerous levels. This layered system, binding proteins in the blood, transporters at the cellular level, and storage in the skin, forms a molecular shield. It is not a single mutation, but a network of defenses, refined by millions of years of evolutionary pressure. Each mechanism works in concert, ensuring that the frog can carry one of nature's deadliest molecules without ever risking its own life. The golden poison frog survives not by changing the rules of chemistry, but by mastering the art of containment. In the tangled food web of Colombia's rainforests, the golden poison frog sits almost untouchable. Its skin advertises danger in a language every predator understands. For most animals, a single bite is a death sentence. Yet nature never stops searching for loopholes. Somewhere in the leaf litter, a slender snake, Limatophis epinephalus, hunts with unusual confidence. Unlike jaguars, birds, or other reptiles, this colubrid snake can feast on the world's deadliest frog and live to hunt again. There is a catch. The snake's resistance is only partial, and its appetite is limited to the frog's juveniles. Even for this specialized predator, the risk is real. Too many toxic meals, and the hunter could become the hunted. This uneasy truce is the product of an evolutionary arms race. The frog's ancestors, once only mildly poisonous, grew brighter and deadlier as generations of predators pushed them to extremes. In response, a handful of snakes evolved subtle biochemical tweaks that shield their nerves from the toxin's onslaught. The result is a standoff. The frog's chemical arsenal keeps almost every threat at bay, but a single codebreaker remains. Each encounter is a high-stakes gamble, with survival measured in micrograms. The battle does not end there. On the far side of the world, in the forests of New Guinea, pitohui birds carry the same batrachotoxin in their feathers and skin. These birds, like the frog, are both toxic and brightly colored, a warning to any would-be predator. Scientists have traced the source of their shared weapon to mellurid beetles, revealing a case of convergent evolution. Two distant lineages, continents apart, have arrived at the same solution: eat the right insects, store their toxins, and wear danger on your skin. Both the frog and the bird have evolved internal defenses, likely specialized proteins and cellular pumps that keep their own nerves safe while turning their bodies into poison vaults. In this chemical arms race, escalation is the rule. The frog's toxins grow stronger, predators adapt or die, and only the most specialized survive. Most of the rainforest's hunters have learned to steer clear. One taste is a lesson enough. But for a select few, the lure of a toxic meal is worth the risk. The golden poison frog's power is not just a barrier, but a challenge, a force that shapes the very structure of its ecosystem. Where most see a dead end, evolution finds a path forward, one adaptation at a time. Deep in the forests of Colombia's Chaco region, the Embera people have mastered a secret known to few outsiders. For generations, they have sought out the golden poison frog, not to eat, but to borrow its power. The process begins at dawn, when hunters slip quietly through the leaf litter, listening for the soft calls that betray the frog's hiding place. Bright skin makes these frogs easy to spot, but only the most skilled can approach. Touching the animal with bare hands is forbidden. Instead, a leaf or wooden stick serves as a barrier, a respect born from hard lessons and old taboos. Once caught, the frog is not harmed. The Embera coax it gently over warm coals or a fire, never letting the flames touch its skin. The heat stirs a milky secretion from the frog's back, thick, glistening, and laced with batrachotoxin. Hunters use slender sticks to collect the toxin, careful not to let it touch their own skin. This is a ritual of patience and precision. The toxin is then smeared onto the tips of blowgun darts or arrows, sometimes mixed with plant resin to make it stick. One frog can prepare enough darts for an entire hunting season. These poison-tipped darts are not just tools, they are a testament to deep ecological knowledge. The Embera know exactly which frogs to seek, which habitats yield the most potent toxin, and how to keep the weapon safe. Darts prepared in this way remain lethal for months, sometimes even years. Stories circulate of a single dart bringing down a monkey or a jaguar with one shot. The danger is real. Only respected hunters are allowed to handle the frogs or the darts, and children are warned never to touch them. Accidents are rare, but never forgotten. This practice is more than survival. It is a living science, passed from one generation to the next, blending observation, respect, and a profound understanding of the rainforest. The golden poison frog, transformed by Embera hands, becomes a bridge between nature's chemistry and human ingenuity, its toxin a weapon forged not in malice, but in partnership with the land. In laboratories around the world, vials labeled batrachotoxin are handled with the care reserved for radioactive material. Batrachotoxin's power is so absolute that even the smallest error can have consequences. No antidote can reverse. Yet for medical researchers, this molecule is not just a threat, but a key, one that opens new ways to study the body's own electrical systems. Batrachotoxin's ability to force sodium channels open has turned it into a precision probe for unraveling the workings of nerves, the heart, and the brain. Scientists use it to map channel structure and to understand diseases where these electrical gates go wrong, like epilepsy and certain heart arrhythmias. The toxin's unique action has inspired the search for new painkillers and heart drugs, with researchers hoping to design safer molecules that mimic its effects without the danger. The promise of turning a lethal compound into a tool for healing is not limited to batrachotoxin. In the early 2000s, a biotech company called Transmolecular developed TM601, a synthetic version of chlorotoxin, a peptide from the deathstalker scorpion. TM601 was designed to seek out and bind to cancer cells, lighting up brain tumors for surgeons and delivering targeted treatments. Early clinical trials showed that the compound could reach tumors deep inside the brain, offering hope for patients with gliomas. But as with many breakthroughs, the path from discovery to medicine proved long and uncertain. Regulatory hurdles, funding challenges, and the complexity of human biology slowed progress, and no chlorotoxin-based drug has yet reached routine clinical use. Chlorotoxin remains a promising lead, but the road to a standard treatment is still ahead. The economics of these molecules are as staggering as their biology. Natural batrachotoxin is so rare and difficult to extract that a single liter, if it could even be collected, would cost tens of millions of dollars. Each wild golden poison frog yields about 1 milligram of toxin, meaning more than 1 million frogs would be needed for a single liter. This is not just impractical, but ecologically impossible. Today, chemists work to build these molecules from scratch in the lab, using painstaking synthetic routes to produce even a few milligrams at a time. For now, the dream of a medicine drawn from the world's deadliest frog remains on the horizon, a reminder that nature's most dangerous weapons may also be its most valuable cures. Right now, golden poison frogs face extinction as rainforests shrink. Yet their toxins hold answers to medical challenges we have not solved. Each disappearing frog erases a unique evolutionary breakthrough, lost before science can catch up. Nature's most dangerous secrets are not just threats, they are opportunities. What we choose to protect today could shape the medicines and mysteries of tomorrow. Thank you for joining us. Please share your thoughts below.